Good afternoon, dear students. I congratulate you on the start of new semester. During the entire semester, we will study medical protozoology in detail, which consists of 3 chapters: medical protozoology, medical helminthology, and medical arachnoentomology. During these classes, we will look at the morphological features of the structure of parasitic organisms, their life cycles, get acquainted with parasitic diseases or infections, and consider methods of diagnosis and preventions. When preparing for classes in a digital educational environment, tests segments for practical classes, as in the last semester, right? As well as an atlas summary will be published. Today we will get acquainted with the topic general and medical protozoology, representatives of the class Flagellates. The lesson is taught by Andrei Pavlov, assistant of the Department General and Molecular Biology. The slide shows the key questions of the topic that will help us learn the topic. Before you start studying parasitology, I suggest you mention outstanding people who became the founders of this science. We are talking about the German zoologist Rudolf Reinhardt and domestic zoologist Evgeny Nikanorovich Pavlovsky. Medical parasitology studies human parasites, the diseases caused by them, develops methods for their diagnosis, treatment, and prevention. Medical parasitology includes 3 sections: medical protozoology, the science of pathogenic protists and human diseases caused by them; medical helminthology, the science of helminths and human diseases caused by them; and medical arachnoentomology, the science of arthropod, mainly ticks and insects, as carriers of pathogenic infections and parasitic diseases of humans. The object of study of medical parasitology in human diseases of parasitic nature, their pathogens and vectors of vector-borne diseases, animal hosts of pathogens of human parasitic diseases, methods and means of diagnosis, treatment and prevention of human diseases. Parasitology studies the phenomenon of parasitism, the biology and ecology of parasites, diseases caused by them. Measures to prevent diseases and ways to control parasites. Parasitism is a universal phenomenon of nature, the most common form of symbiosis. Parasites are all viruses, many bacteria, some types of fungi and higher plants. For example, there are 10,000 known species of parasitic protists. In the animal world, parasites include 7,000 species of arthropods, 20,000 types of helminths. Some classes are entirely represented by parasitic organisms. There are spores, suckers, and tapeworms. You do not have the parasitic representatives of the only types of sponges, colenterus, and echinoderms. Parasites, according to Pavlovsky, are organisms that live at the expense of individuals of another species, being biologically or ecologically closely related to them in their life cycle for more or less of its late. The relationship between the parasite or the host as well as the relationship in the predator-prey system is subject to certain ecological laws. Parasites that take part in the regulation of the host population and sometimes determine the direction of microevolutionary processes. According to Pavlovsky, habitat of parasites dual environment first order, the immediate habitat of the parasite in the host organism, and environment second order, the physical, chemical, and biological parameters of the habitat surrounding the host external environment. Parasitism can be classified in four ways. By the similarity of relationships by the closeness of the relationship, by time, and by localization. The life cycle of a parasite is totally of all the stages of the parasite's pathogenesis and the path of its transmission from one host to another. It is cyclical in nature. First, invasive stages for different hosts. Second, methods of infection. Third, features of reproduction. Fourth, exit gate from the host body. Fifth, final and intermediate hosts. Sixth, free living stages, and seventh, resting stages. Ways of infection with parasitic diseases. Alimentary. Third, the mouth, often with food and water. Airborne. Respiratory. Through the mucous membranes of the respiratory tract. Contact household. Direct contact with a sick person of animal through underwear and household items. Vector-borne or the blood-sucking pernostation. A. Inoculation through the proboscis carrier in the blood. For example, malarial plasmodium and trypanosome. And B. Contamination. Transplant placental via the placenta. Percutaneous through the skin. Sexual during sexual contact. Transfusion and during transfusion of infected blood and organ and tissue transplantation, and artificial. Depending of the stage of the development, the parasite, the host are definitive, basic, final. In their body, the parasite reaches sexual maturity and passes its sexual reproduction. For example, a person for an armed tapeworm and malarial mosquito for malaria pathogens. Intermediate, the larval stage of the parasite lives in the body or passes through its asexual reproduction. For example, a pig for an armed tapeworm, a person for malaria pathogens. Additional or second intermediate hosts for example, fish for a cat sucker. A reservoir in the body, there is an accumulation of invasive stages of the parasite without its development. For example, a predatory fish for the broad sloths, wild rodents for leishmania. Depending on the conditions for the development of the parasite, the host can be obligate, provide optional conditions for the development of the parasite. The best survival, rapid growth, the greatest fecundity. Hence, there are biothenotic connections, the possibility of natural methods of infection, and good biochemical conditions. For example, a person for human Ascaris, and broad lived sloth. Facultative, characterized by the presence of biothenotic connections, but the lack of optional biochemical conditions for the development of the parasite, so their body shortens its lifespan, or it doesn't pass the full cycle of development. For example, a cat for a broad sloth, or a person for a pig Ascaris. Potential provide the biochemical conditions for the development of the parasite, but there are no biothenotic connections. Natural routes of infection, for example, herbivores for trichinella. The concept of pathogenicity, parasitic diseases. Pathogenicity, the ability of the pathogen to cause diseases in animals of a certain species or in humans. Virulence, a measure of pathogenicity. Carriage, the absence of symptoms in pathogenicity of the parasite in known. For example, cytosilicate for Entamoeba histolytica. Factors affecting pathogenicity. Host genotype, age of the host. Young organisms are not sensitive to infection. The food regimes the host and inferior diet weakens the body of and contributes to an increase in the number of parasites and the number of eggs they lay. The presence of other parasites and diseases in the host. Parasite genotype. There are highly pathogenic and weakly pathogenic strains of parasites, for example, bacteria. The profession of our teniasis and beef tiform infection usually become infected workers of the meat. The Diphyllobotrys fishermen and place of residence of the host. Helminth infections are more common in rural residents. Pathogenic effect of the parasite. Medical manifests itself in the form of tissue damage by sexually mature forms, the organs of attachment with hooks, suction slots, suckers, part of the oral apparatus, larval stages when the larva of blood flux penetrate through the intact skin. Eggs then that have spines and damage the walls of blood vessels and the mucous membrane of the bladder of intestines. The accumulation of eggs causes cirrhosis of the liver, a blockage of the intestinal lumen, and even a rupture of sexually mature forms as carries a broad lentil. Pathogenic effect of the parasite. Toxic, the effect of the parasites waste products on the host body. Malarial plasmodia, the release of the dissimulation products from red blood cells into plasma causes fever attacks in the patient. Helminth can be a cause low blood pressure, shortness of breath, inhibition of blood clotting, loss of appetite, weight loss, anemia, Crisia, fatigue, insomnia, nausea, vomiting, diarrhea, and delay not only physical but also mental development. Pathogenic effect of the parasite. Stealing from the host when feeding the parasite. Tissue, fluid, tissue, blood, and processed food. Damaging effect during the migration of larval forms through the body. Destruction of tissues, ascaris, inflammatory process, contribute to the penetration of infection, toxic effect, and the spread of pathogenic of infectious and invasive diseases is carried out by blood-sucking ectoparasites, lice, fleas, ticks. The action of the host of the parasite. The host organism also affect the parasite. Its action is aimed at suppressing the vital activity of destroying the latter. The responses are divided into local and common. Local is cellular reaction and tissue reactions. Common humoral reaction is immunological reactions of the production of protective specific antibodies in response to the arrival of antigens released by the parasite. The appearance of immunity, permanent and temporary. Human parasitic diseases are a separate group of diseases. All of them are provoked by parasitic unicellular, uncellular, and unicellular organisms. The most common parasites are the worms, helminths, followed by arthropods, insects, and ticks. Parasitic diseases are called invasive diseases or simply infestations. The main thing of parasitic invasion are allergic reaction by the type of urticaria, increases body temperature, lymphadenitis, arthralgia, and myalgia. Pulmonary syndrome often occurs and manifests itself in the debilitation, tating, cold shortness of breath, chest pain, abdominal syndrome, neurotic disorders, skin diseases, disorders of the genitourinary and excretory systems, and frequent colds. Parasitic diseases or parasitosis, depending of the nature of the pathogen, are divided into several groups. Infection, mycosis, uh, invasion, and infestations. Classification of parasitic diseases or parasitosis. Depending on the range of host zoonoses, are diseases peculiar only to animals. Pathogens are transmitted from animal to animal. Anthropozoonosis are diseases peculiar only to human. Pathogens are transmitted from person to person. And anthropozoonosis diseases whose pathogens can affect both human and animals. Depending on the pathogen. Protozoologies, pathogens of protists, helminthiasis, the causative agents of parasitic worms, echerinosis, pathogenic of mites, infections, insect pathogens, disease outbreaks in human population are called epidemics in population of animals of the epidemic. Prevention by parasitic diseases of reduced to compliance with the following recommendations. Compliance with the rules of personal hygiene. This applies to washing your hands before eating, after each visit to the toilet or restroom, public places, and after contact with animals. All food products must be properly processed. Meat and fish must be treated thermally. Water should be boiled. It is necessary to conduct preventive antiparasitic courses for pets. Regular wet cleaning should be carried out the room. When working with the ground, the hands should be gloved. There are the main measures for the prevention of parasitic diseases, which will allow you to protect yourself and your loved ones from possible invasion as much as possible. General characteristics of the protozoan type. There are organisms whose body consists of cytoplasm and one or more nuclei. The protozoan cell is an independent individual that performs all the functions of a complete organism. The vast majority of protozoa have microscopic dimensions, passing through complex development cycles. The parts of the protozoan body then performs various functions are called organelles, which are of two types: general purpose and special purpose. And organelles of cells regulation, excretion, and respiration. In protoplasm of protozoa, two layers can be distinguished. Ectoplasm, located on the outside, in its dense and homogeneous. From it, a peripheral film informed pellicle on the surface of which sometimes a cuticle is formed and study the table below and write down the characteristics of the flagellate class in your notebooks. Before we start studying representatives of the flagellate class, we need to learn taxonomy with an English and Latin name. So, taxonomy of flagellates. Type: Protozoa. Subtype Plasmodroma. Class: Flagellata. Mastigophora. Squid. Polyfluidaceae. Polymystigina. Type: Lamblia intestinalis. Trichomonas vaginalis. Trichomonas hominis and Trichomonas tenax. Flagellates are widely distributed in fresh water bodies and as well as the seas. Many species are parasitic in various animals and humans, and thus cause great harm. Trypanosomes, human intestinal parasites. The intestines of vertebrates, as well as humans, the habitat of many species of heterotrophic flagellates. Complete the suggest tasks in the atlas. On this slide, it is necessary to correctly position of the host factors of the parasite. And the next slide to learn anatomical and morphological features of Lamblia intestinalis. Pay attention to the video which shows giardia under a microscope. Giardia is a common infectious diseases of the human body caused by the intestinal single-celled parasite Giardia. Giardiasis occurs in adults and children all over the world. The main method of infection with giardiasis is fecal-oral. Cysts with human or animal feces enter the internal environment. They are able to remain viable in water bodies for several months. The causative agents of giardians can withstand freezing, increasing the temperature to 50 cells. They are killed by boiling. Infection with giardias is possible when drinking tap water, when washing fruits, when vegetables with employed water in the summer. You can get infected by swimming in a pond infected with giardia cyst. Classification: giardiasis, asymptomatic giardiasis. There are no signs of the disease. but the person release giardiasis into the environment. The infectional form is a violation of the functions of the intestine. Hepatobiliary form a violation of the function of the liver bile ducts. Giardiasis is a concomitant diseases when the main chronic disease, atopic dermatitis, skin allergy, celiac disease, a genetic disease in which the protein gluten causes damage to the small intestine mucosa, and the absorption process of disrupted, causes decreases in immunity and various infections such as giardiasis are attacked. Giardia is difficult to diagnose without laboratory confirmation. Giardiasis is suspected. It is sent to a blood test, fecal examination, and duodenum. The most accessible method of laboratory diagnosis of giardiasis of the coprological examination. However, it showed the note the giardiasis in the feces cannot always be detected. In addition, it is advisable to conduct a study of the duodenal content. A serological diagnosis of giardiasis is also performed when specific antibodies are detected in the blood 2 or 4 weeks after infection. Prevention of giardia infections include a set of standard measures recommended to protect hygiene. Infections with a fecal-oral transmission mechanism do not drink raw water, do not swallow water while swimming in open water, and explain to children that this should not be done. Thirdly, wash all fruits and vegetables served on the table, as well as your hands before eating. Do not allow pets to lick your face and hands, and are required to same treatment of animals from children. It is much easier to prevent the invasion than to carry out the subsequent treatment of giardiasis. Complete the task in the atlas. Specify the definitive morphological features of urogenital and intestinal trichomonas. Make the appropriate notation and list them. What diseases do they cause? Cyst form if the form data. Pay attention the video which shows trichomonas under a microscope. Pathogenesis. Orogenital trichomonads have a primary pathogenicity and are able to cause and obvious or hidden infection in all people. There is no innate immunity to them, but some having humans infection leads only to short-term in men. Trichomonas can parasite in the urethra, parauretral passages, the preputial sac, the appendages of the testicles, and the accessory sex glands in women in the urethra, the glands of the vestibule, the vagina, the cervical canal. The rare cases trichomonas enter the uterine cavity or cause and extending process. There is no hematogenic dissemination, thus also several lesions usually appear in trichomonas infestations. They are all limited to the genitourinary system. However, trichomonas invasion is accompanied by the appearance of various antibodies in the blood serum, which do not provide any noticeable protective effect. Reinfection with this infection is possible. Pathogenicity of oral trichomonas. It is considered conditionally pathogenic. Also, there is evidence of a much more frequent occurrence in people with various diseases of the oral cavity and teeth, gingivitis, periodontal diseases, dental caries, and the unfavorable role of the protozoa and maintaining the pathological process in the oral cavity. Trichomoniasis is a lesion of the genitourinary organs, which belongs to a group of sexually transmitted diseases. Diagnosis is based on the detection of trichomonas in the secretion from the urethra. In the light microscope, browsing native drugs urethral discharge, sediment, newly issued centrifuged urine, prostate secretion, which allows you to observe movements of trichomonas against stationary leukocytes and epithelial cells. Dried and fixed smears from the urethra are stained with methylene blue or gram. But in this case, it is difficult to distinguish the trichomonas from the thyroidine epithelial cells. Special coloring techniques are required to identify them. In the diagnosis of urogenital trichomonasis, of the cultivation of trichomonas on special nutrient media is also used. The method of solving is recommended for use if an asymptomatic latent form of trichomonasis is suspected. Apply the accelerated RIF 40 as a selection test and intradermal test with vaginal trichomonas with proposed. Rapid screening allows for a micro precipitation reaction with latex particle and antibodies to the pathogen to determine the Trichomonas vaginalis antigen in separated genitals. The method of selective but the diagnostic identification of anterior of moralnic antibodies in the theorem also applies to auxiliary methods to research. On this slide, show Trichomonas strained with methylene blue and Trichomonas colored gram. Prevention orogenital trichomonas. Until complete cure, the persistent disappearance of pathogens, patients are prohibited from having sex without a barrier means of protection. Persons who have had sexual contact with the patient must be identified and involved in treatment. Control examinations, smears and cultures, urological and gynecological examinations with the use of elementary and mechanical provocation begin 7-10 days after the end of treatment. Complete task in the atlas. Table number 3. Life cycle of Leishmania. Describe the life cycle of Leishmania below. Specify the stages of development of the parasite. What is the clinical picture and pathogenesis of the disease caused by this parasite. Cutaneous leishmaniasis is the infection disease caused by a protozoa and accompanied by damage of the skin, less often the mucous membranes. The most common species are Leishmania tropica, Leishmania major, Leishmania aetiopica, Leishmania infantum, Leishmania mexicana, Leishmania amazonensis, and Leishmania brasiliensis. Can you see this picture and consider the presented image in the workbook? Make the notation. Diagnosis have 4 methods: physical examination, laboratory tests, detection of infection agents, and instrumental methods. Specific prevention of indicated for everyone traveling to endemic areas. It is recommended to carry out vaccination in the winter-autumn season and no later than 3 months before the planned trip. The main means of non-specific prevention and vector control, drainage of basements and vegetable stores, insecticidal treatment, the use of repellents, timely diagnosis, isolation and treatment of patients, and the installation of mosquito nets in the wards, they are important. We recommend routine vaccination of domestic dogs and fight against urban landfills, desertization, and shooting of sick wild animals. Visceral leishmaniasis is a chronic infections, protozoal diseases which damage of the liver and spleen, the development of anemia and cachexia. Visceral leishmaniasis have 4 methods diagnosis. Physical examination, laboratory tests, detection of infection agents, and instrumental methods. Specific prevention consists in the planned introduction of a live vaccine recommended for people living or planning a long or short stay in endemic areas, tourists. The drug should be administered in winter or autumn at least 3 months before the trip. Non-specific measures to prevent visceral leishmaniasis include timely detection, isolation, and treatment of patients. The use of mosquito nets, vaccination of dogs, elimination of handfills, disinsection and desertization measures. The fight against mosquitoes consists, among other things, in draining swamps, basement and vegetable storehouses. Chagas disease is a vector-borne protozoan infection, the causative agent of which in the pathogenic Toxoplasma cruzi, and the carrier of triatoma bugs. Complete tasks in the atlas. Table number 4 define a representative of the simplest type. In the laboratory, the following blood test results were obtained. Trypanosomes were found in the smears of the blood taken shows in the figure. Name a specific type who is the carrier of causative agent of the disease. Does it pose an epidemiological risk? And if so, what preventive measures should be taken? Diagnosis of Chagas disease. Diagnosis of Chagas disease includes a third collection of anamnesis, analyzes the clinical picture and laboratory studies. Blood cerebrospinal fluid punctates from the primary sites of the lesion, lymph nodes, spleen. The bone marrow are used as a test material. In the actual stage of Chagas disease, the first 6 or 20 or 12 weeks. Parasites can be detected by microscopy of a thick drop of blood or fixed colored preparations. Bacteriological culture of the patient's blood helps to identify the picture culture of Trypanosoma cruzi. In Chagas disease, xenodiagnostics is used, which includes the study of intestinal contents of an union-involved triatomic bug after it is saturated with the patient's blood. It is possible to conduct biological tests with the introduction of the patient's blood into junior pigs or wild mice, and then study the tissue samples. In chronic Chagas disease, serodiagnostics are more effective. They complement binding reaction, the indirect fluorescence reaction, the indirect hemagglutination reaction, and ELISA. The presence of specific LGM indicated and acute stage of Chagas disease with chronic LGG only. Some infections such as malaria, syphilis, and leishmaniasis can give false positive results for trypanosomiasis. So it is recommended to use at least two independent serodiagnostic methods. Differential diagnosis of pathogenic trypanosomes from non-pathogenic species that can be found in the human body without causing the development of the disease is necessary. Depending on the geographical area, World Health Organization recommends the following prevention and control approaches. Spraying of house and surrounding areas with residual insecticides, modernizing homes and maintaining cleanliness in the home to prevent infection with disease vectors. Token individual preventive measures such as the use of bed nets. Observe proper hygiene measures in the preparation, transport, storage, and consumption of food. Screening of blood donors, testing of organ, tissue, and cells, donors, and recipients. Ensuring access to diagnosis and treatment for persons who are medically indicated or recommended for antiparasitic treatment, especially children and women of childbearing age before pregnancy, and screening newborns and older children both to infected mothers who have not received antiparasitic treatment to ensure timely diagnosis and treatment.